In this lecture, we're going to learn the following topics: the definition of anaerobic metabolism, phosphocreatine system, phosphocreatine reaction equation, law of mass action, glycolysis, including substrates, important enzymes, products, and the fates of pyruvate. ATP is needed for numerous functions inside the cell. For example, the synthesis of new proteins when old ones become damaged and need to be replaced. For active transport, and we talk about ATPases and about the use of ATP to transport substances in and out of the cell. ATP is also used for movement, and we talk about the various places where ATP is used to contraction and relaxation, cross-bridge cycling, myosin ATPase, sarcopumps ATPase. So the body uses different fuel sources, for example, carbs, fats, and protein, and breaks them down and uses them to generate ATP. So this process is known as metabolism. So the metabolism is a sum of the biochemical reactions involved in maintaining the living state of the cell. And we can classify metabolism in two types of reactions. We have endergonic reactions, which consume ATP for muscle contraction, synthesis of new proteins or molecules in the body, and for active transport. And we have exergonic reactions that that make the breakdown of fuel sources, such as glucose, to produce ATP. And the breaking of these bonds releases energy, and we're gonna see this how this works better later. So the metabolism in the body can be divided in two categories: anaerobic metabolism and aerobic metabolism. Anaerobic metabolism, it's energy generating system without the presence of oxygen. So in the anaerobic metabolism, for example, in oxygen-independent metabolism, typically takes longer to generate force via aerobic pathways. And in these cases, anaerobic metabolism is used to generate ATP quickly until aerobic systems kicks in. And then we have aerobic metabolism, which is energy generating system under the presence of oxygen. So aerobic means oxygen dependent, and aerobic metabolism refers to an energy generated system under the presence of oxygen. And the substrate is the chemical of interest that is being modified. So in biochemistry, an enzyme substrate is a material upon which an enzyme acts. So as substrates, we have carbohydrates, fats, and proteins. So those are the substrates or the materials required to generate ATP. And we will see how enzymes act on these substrates to convert them into energy, ATP, that can be used by the cell. A few examples of endergonic reactions. So we have endergonic reactions for mechanical work of muscle action. So for muscle contraction, we talk about how ATP is utilized during contraction. Myosin ATPase allows myosin head to break down ATP and perform power stroke and pull acting, causing muscle to shorten and generate force. We also have chemical work involving synthesis of cellular molecules. So converting various substrates into bigger molecules, glucose and glycogen, for example, glycerol and fat acids to triglycerides, which can be broken down for energy when needed. Using individual amino acids to make proteins is another example. Building blocks for muscle and various other tissues, it's another example. And then we have transport work involving active transport of substances, bringing various ions in and out of the cell, which allows for generation of action potentials, For example, circuit pumps. So ATP, adenosine triphosphate. So let's take a close look at an ATP molecule. So an ATP molecule, it's formed by adenine, ribols, and triphosphates. That's why it's called adenosine triphosphate. It's connected by 3 high energy phosphate bonds. So the breaking of ATP bonds is an exergonic reaction. When broken, releases energy. And in this case, ADP in this image, it's adenosine diphosphate. So cells use this energy to perform various forms of work that we'll look at on this until the last slide of this lecture. So a free inorganic phosphate molecules can combine with ADP, adenosine diphosphate, which is 2 phosphate molecules, to reform ATP. So a combination of ATP with inorganic phosphate can reform ATP. And energy required to reforming these bonds comes from breaking down of various fuel sources such as carbs, fats, and proteins. So according to law of conservation of energy, energy can never be created or destroyed. It can only change forms. So the term substrate level phosphorylation refers to the synthesis of ATP by reactions in which ADP is one of several substrates and ATP is one of several products of an enzyme catalyzed reaction. So the anaerobic sources of ATP that we're going to look over this lecture are phosphokeratin system and glycolysis. So in the anaerobic metabolism, provides energy much faster than oxidative phosphorylation or aerobic metabolism, which requires the presence of oxygen to generate ATP. So the anaerobic metabolism comprised of phosphokeratin and glycolytic systems allow for substrate level phosphorylation and provide an immediate source of energy. Starting with phosphokeratin system. Phosphokeratin, also known as creatine phosphate, it's a phosphate group attached to a creatine molecule. This system provides rapid energy, so it's a source of rapid energy, and phosphokeratin can anaerobically donate a phosphate group to ADP to form ATP during the first few seconds following an intense effort, for example, sprinting or weightlifting. So 1 molecule of phosphokeratin generates 1 molecule of ATP. But the excess of ATP can be used during a period of low effort to convert creatine to phosphokeratin. The reversible phosphorylation of creatine, for example, both the forward and the backward reaction is catalyzed by creatine kinase. So creatine kinase, it's the enzyme responsible for catalyzing this reaction. And in this case, those enzymes are there. They have catalyst function. So catalyst, because it's a substance that increases the rate of a chemical reaction without itself undergoing any permanent chemical change. So creatine supplementation started in the 90s and has been used until nowadays. Athletes believe that the supplements made them bigger, stronger, and faster with minimal side effects. However, studies also indicate that the degree of increase varies considerably between individuals, possibly because of differences in the subjects that are being tested in the studies. The evidence regarding whether an increase in the body's creatine levels translates to improved muscle activity is inconclusive. Although the the role of creatine supplementation is still being studied, there is a lot of people that still uses creatine supplementation. So phosphokeratin system, um, in this image, we can see that ATP levels are maintained during intense exercise via the breakdown of phosphokeratin via the creatine kinase reaction. The inorganic phosphate that is released can combine with ADP to form ATP. So depleted relative quickly, therefore it's an immediate source of energy. So in this graph, we see that the changes in phosphokeratin and ATP during a 4-second maximal sprint, and during the first 8 seconds of exercise, ATP levels are maintained. And as the phosphokeratin pool is used up, it can no longer replenish ATP as fast as it is consumed. So that means that this phosphokeratin is depleted relatively quickly, therefore it's an immediate source of energy. So in terms of law of mass action, let's take a closer look at the regulating of the phosphokeratin system. So to understand how this reaction works, let's take a look in this law of mass action. The rate of chemical reaction is directly proportional to the concentrations of reactants or substrate. Remember that flow is equal to delta P times conductance. So in this PCR reaction flow, we have delta P, which is the concentration of substrate, in this case, the reactants and our products. We have the conductance, which is the activity of creatine kinase enzyme. And according to the law of mass action, if we increase substrate, we will increase flow by driving this reaction forward.